firstly a great opportunity to present to you MMB, and like about half of you are in the tutorial today, more than half probably. The rest of you can think about using MMB. So I'm going to start with an overview. The goal of this project was to model the trajectory of ribosomal translation as tRNA starts in the classical state, hybridizes, and then completes the translocation. That's part of a larger goal of producing the entire cycle of ribosomal translation at all atoms resolution. So I'm going to start with an introduction of MMB, or macromolecule builder. Some of you that are in my tutorial have already seen a lot of this, so I'll probably be a little bit fast about that. Then I'm going to talk to you about the constraints that are available from various pieces of experimental data on the ribosome, and most importantly, these cryo-EM maps from the Stark lab. Lastly, I'll show you the predicted trajectory of translocation, and actually I'll probably skip the part about FUSB. So here's sort of an overview of what's available in terms of structures of the ribosome. There are lots of 3D structures solved by crystallography. There are more structures that are available only at low resolution, for example, by cryo-EM experiments. But there's no good way up to now to connect these static structures into a dynamic picture of the trajectory. So we might be missing a lot of crucial details about how things happen, how the tRNA lets go of one site, shifts to another site, and jumps over the various barriers it finds along the way. And so I'm going to show you what I've done to remedy at least part of that. So again, I'm focusing just on the elongation cycle right here, going from classical to hybrid and then completing the translocation. So this is sort of a broad overview of the sorts of things that are going on. You start with the classical state. Elongation factor G comes in. Sometimes it gets stalled by fusidic acid and antibiotic. FUSB comes in and unblocks it, and if that's happened. EFG comes off after helping the tRNA translocate. The tRNA gets sort of helped out of the E site by the L1 stalk. And then another tRNA can come in bound to EFTU. Again, I'm focusing on the movement of these tRNAs as they go from classical to post-translocation states. So first, a brief overview of macromolecule builder. MMB works in internal coordinates, and I'm going to show you what it means and why that gives us some advantages. Then I'm going to talk to you about the constraints and forces that you can apply to your system and how you can control its flexibility. I'm talking a little bit about this feature called physics where you want it, which I don't think you saw in the tutorial. And then I'm going to talk to you about the density-based force field. And that's the first part of my talk, which is the technical part. So those of you who are in my tutorial know that in MD, the various, all the atoms in your system have three Cartesian degrees of freedom. So your total degrees of freedom are 3n. In internal coordinates, things are set up differently. Here you have a root atom, and all other atoms in the system are dependent on that root atom in an open tree structure. And the atoms can be connected in one of several ways. The most typical way is by these torsion mobilizers, which can have a fixed bond length, fixed bond angle, and all it can change is their dihedral angle or their torsion angle. That's the kind of mobilizer you have on the backbone in most of the ribos. You can also have this so-called rigid mobilizer, which has no degrees of freedom. So two atoms that are connected by this mobilizer are one body. And I don't mean that they're effectively one body, I mean that they are one body. This entire base is composed entirely of rigid mobilizers, therefore it has exactly 1 degree of freedom. And so it costs as much to simulate as a single atom would. Lastly, there are free bonds which can change their bond length, angle, and dihedral. And the cool thing about MMB is that you can decide for any stretch of residues that you want to leave it at this default bond mobility, or you want to change it all to rigid, or all to torsion, or all to free. So if you use MMB, you're going to find yourself rigidifying all kinds of things, because that way you can take a gigantic complex with lots of degrees of freedom, identify domains that you think are stable and they're not going to move much, rigidify them, and reduce the degrees of freedom down to maybe a few dozen, maybe a few hundred, and that way your computation time goes down to something very small. Okay, so one feature which I used in this study is called physics where you want it, and as you know, molecular dynamics costs you a lot if you're dealing with a large complex, because you have to calculate so many non-bonded interactions, electrostatic and van der Waals. So with physics where you want it, you can limit yourself to a specific region of interest. And the way that this works is you specify, let's say you're interested in what happens, this rearrangement when one residue is mutated at this antibody-antigen interface. You can flexibilize just 5 residues, let's say the mutated residue and 2 residues to the left and right. And then you can turn on the physics, the electrostatics and the van der Waals interactions for additional residues within some envelope, let's say within 10 angstroms of the flexible residues. And that way, and the rest of this complex, for example, all this part of the antibody that's out here, this part of the antigen that's out here, is rigid, fixed to ground, and non-interacting. And so again, now you've turned many thousands of degrees of freedom and probably many thousands of forces into just a few dozen degrees, a couple dozen degrees of freedom, and then a very relatively small number of forces. And so this will converge very quickly, not only for those reasons, but also because of 2 additional reasons. Number one, if you're working in internal coordinates, it's a lot harder for your system to explode. Let's say you have a system like this, where you turn, let's say an alanine into a tryptophan, like a massive change in volume, and you want to equilibrate. You want to know where this tryptophan is going to go to where it's no longer clashing. It's in a more comfortable position. In MD, this thing would just explode, right? You have all these close contacts. The atoms will fly off into deep space. You wouldn't even be able to minimize it. So, but in internal coordinates, the sort of these exploding degrees of freedom simply don't exist because, first of all, let's say this residue, maybe the whole side chain could be rigid, or maybe they're all torsion bonds. In any case, they can't change their bond length. And there's a second reason that it's more likely to converge, is that we have what's called a variable time step integrator. And that means we can make the time steps really tiny. The time steps are not fixed. So if it needs to take a shorter time step to calculate, to resolve some very large forces, then it can do that. It can make them arbitrarily small, and then it'll take longer, but it's more likely to converge. So actually, you can see here what happened when we turned on the calculated dynamics of this. It as I mentioned, it was able to equilibrate the new and find the new position of this tryptophan. We also have a density-based force field, and again, I apologize to those of you who heard this earlier. But the way this works is it's very similar to a program written by Klaus Schulten called MDFF. In MDFF, you have a conventional molecular dynamics force field, and on top of that, you have a potential that's sort of trying to guide this molecule into this density map that you provide. So let's say you have a density map at low resolution, and you have the crystallographically observed structure from, let's say, some other organism or in a different conformation. What happens is that these forces, which are proportional to the gradient of the density at the nuclear position, are all going to try to sort of push these atoms into regions of higher density. And so at the end of some equilibration time, this flexible protein will be fitted approximately into this density map. And so this is a very popular method, and it works really well. The only problem is that, as you might imagine, for something this huge, to run MD takes a lot of computer power, at least by the standards of, let's say, an experimentalist or a computational person that's not exactly an MD specialist. For example, the 16S subunit of the ribosome takes about 48 processors in about 5 hours. So what I did was I simplify this problem. I said, well, I can identify certain domains that are stable, and I can rigidify them. So just remove all their internal degrees of freedom and just leave flexible this hinge region. Further, I don't really need to calculate the physics within the domains because those are, like I say, rigid. So maybe I just want to calculate the physics at the interface and around the hinge region. So if I do that, not only have I reduced the forces, I've also reduced the degrees of freedom. And at the end of the day, I get a very similar fitting, except it takes probably 2 orders of magnitude less computer time. So you can do it on your laptop and it'll converge in about half an hour. Speaking of that's for the case of 16S. So actually MMB, which was formerly called RNA Builder, is available at simtk.org, and it's free. You can download it and run it on Mac, Windows, and Linux. And it's got a tutorial that can step you through some simple exercises to help you learn. Okay, so that's it for the technical part, and now we're moving on to the scientific part of my talk, in which I'll tell you how I fitted the ribosome into these various density maps and what we learned from that. So first, I'm going to tell you about the various constraints that can be called from the literature and how we can impose them in MMB. First, we need to talk a little bit about the flexibility of 16S. So 16S has a body domain and a head domain, and it's connected by this thing called the neck. And those of you who worked on the ribosome will probably not debate the fact that the neck is flexible. It's very documented by many crystallographic structures, by flexibility prediction algorithms, etc. So what I did is I flexibilized the neck, but there's an additional constraint that's less well known. There's a sort of a tetra loop that comes out underneath the head and makes some base pairing interactions with the body. And so there are actually 2 points of contact connecting the head and the body. And so you can draw a line between those 2 points of contact and realize that there's a single axis hinge. So there's really basically one degree of freedom. This hinge that connects head and body is basically a 1 degree of freedom hinge. And it's just going to swivel around that hinge like a door, just like a door would. And further, I put some steric spheres on this. I didn't mention these, but there's a very cheap way of making sure that atoms don't crash into each other. And so if I do that, I'm able to explore all thermally accessible conformations of the head with respect to the body in about, let's say, 30 seconds of computer time. And you can see that this trajectory comes within less than 2 angstroms of all 3 observed states of 16S. The 16S in the classical state, the hybrid state, and an intermediate that was crystallized by Jamie Kate. So that's what you're looking at here. You start out with this R2 structure, and it comes back and it comes within 1 angstrom of the R2 again repeatedly, and then it also comes within 2 angstroms of the R1 structure, the classical state. You see here, here, and here. And so it samples all 3 of these states, like I say, several times within within 30 seconds. So that tells us that it's a very efficient way of sampling alternate conformations. So another constraint that we had was this base pairing interaction between the tRNA and the head. This was predicted by Shoji et al. From phylogenetic alignments. And we observed it also in a previous paper where we fitted an all-atom structure to the density map of the hybrid state, and it had already been observed in the classical and intermediate states. So we're absolutely sure that this thing remains in contact throughout the motion. So we can apply a base pairing force, keeping these 2 residues together, and that gives us an additional sort of constraint. It's not technically a constraint because it's a force, but what I'm trying to say is that it'll guide the trajectory. And most importantly, we have these 8 density maps from the Stark lab, where what they did was they did some they started the ribosome in the post-translocated state and looked at the back translocation and observed and clustered several states along the back translocation trajectory, and they sort of postulated that the back translocation should be the reverse of the forward translocation trajectory. And so this gave us a really nice view of what the ribosome does, going from classical to hybrid and then to the post-translocated states. So we started out by fitting to this density map and then progressively fitting to the other density maps. So we have the density maps. We have these interactions between, like we know the base pairing interactions between the tRNAs and 23S. We have the single axis hinge model for the 16S that we described earlier, and this contact between tRNA and the 16S head. And again, these 8 intermediate states at low resolution. So we're going to combine all this data, like I say, by using this semi-flexible fitting algorithm. And we found a few interesting things. First, we illustrated this sort of ridge, helix 78, between the P and E sites, which looks to be very important in terms of determining when tRNA can go from the P to the E site. And more interestingly, we also found a possible mechanism for jumping over this gate. So helix 24 and helix 29 in 16S are too close together for the tRNA anticodon stem to fit through. See, this distance is too small. So there's been a lot of talk about how they should go over. Like maybe the head would open up, so this thing would go to the right and widen the channel. Jamie Cade actually proposed that it worked like a pawl, like there's a sort of like a gear, and pawl is like a ratchet mechanism. And he proposed that the head would sort of go down and let the tRNA go through it and come back up. So our model suggests that the opposite happens, that tRNA actually hops over the gate and then settles in once the gate has passed. Here's another illustration of the same mechanism. Oops. See, here's the gate going past the tRNA. tRNA sort of has to lean a little bit and then go over the gate and settle back down. So it'll be interesting if this trajectory can be used to pull additional data out of additional sort of clusters out of the density maps that were collected in the Stark lab. And this is again another close-up of this process as the tRNA goes over the gate and settles in the post-translocation state. There are a lot of things that you can glean from looking at an all-atom trajectory of the translocation that you wouldn't be able to get from looking at static structures. For instance, we haven't actually fully characterized this in this figure, but you can see that there's not very much net motion. You see there's a lot of things moving around. This is basically a time lapse of the whole process of translocation. You can see that there's a lot of motion of the tRNA, but the anticodon stem is actually not moving all that much in net. A lot of the motion is the 16S moving out underneath the tRNA and letting the tRNA settle back into its new location without moving so much from its original position. And so here's a trajectory of the entire cycle of translocation. You can see here's the post-translocation, and then the yellow one helping the tRNA eject. So and this trajectory is downloadable. Or in other words, we're going to distribute it with our paper, which should be out reasonably soon. So, oops, actually, I need to update some of these statistics. Sorry, but at any rate, the steps from one density map to the next varied in computer time, but most of them took roughly about a day. Whereas with other methods, it's simply not possible to generate the trajectory. What is possible is for you to do things like that. I'm running this tutorial. It takes about 2.5, maybe 3 hours to do the basic part. And I think it's relatively easy for non-specialists to learn. And again, it's distributed for free. And I run lots of workshops, including this one. So the conclusions about the elongation trajectory is that, again, the 16S neck is considerably less flexible than even the single axis hinge model. Sorry, I didn't show you this. But the actual range of motion that we observed in the structures is a lot less in terms of the angular rotation than this model will explore. Again, I didn't go into this in great depth, but the translocation is a cooperative motion between tRNA and the 16S. The tRNA again is moving a little bit less than you might expect just from looking at these structures separately. Let's see. The most significant steric barrier between the 23S P and E sites is this ridge formed by helix H78. This has implications as to when exactly the hybridization happens. Let's see. And I showed you this sort of Paul mechanism where the tRNA acceptor end hopped from the P to the E site by going over the gate formed by the helices H24 and 29. And lastly, we produced the first all atoms kinematic trajectory of tRNA translocation in the ribosome. And we've shown that doing semi-flexible fitting in internal coordinates provides accurate fitting. Actually, I didn't show that particular comparison, but it gets very similar results to MDFF, except again at maybe 2 orders of magnitude less computer time. I have about a year's worth of funding for a postdoc if anybody's interested in this sort of thing, and we're working on getting more funds. And I would like to acknowledge Michael Sherman, who is the lead software architect of Symbios. Symbios is the center that wrote the internal coordinate mechanics engine that underlies MMB. And I was in Symbios for 3 years, and that's where I wrote my software. I'd like to acknowledge Maria Selmer, a ribosome crystallographer at Uppsala, who's been very helpful in this project. Ruben Gonzalez, who criticized very helpfully an earlier an earlier manuscript, and Johan Åkvist, the head of my program, who has also been helpful on ribosome matters. And with that, I'd like to turn it over for questions.
Okay. Any questions here?
Is it possible to perform docking experiment using MMB without electron density modeling?
It depends on what you mean.
Predict the structure of the components.
You mean without for example, without knowing where the interfaces are?
Let's say without.
Yeah, without it, probably not. I mean, I could see how if you had an idea about how to make a docking algorithm, you could pretty quickly implement it in MMB. But no, there's no de novo docking available. What I have done is combine experimental data. Like let's say you know which residues are interacting. You can basically sort of apply these springs to the two subunits and bring them together that way. You can turn on physics at the interface and equilibrate them a lot faster than you could, for example, in MD. That sort of thing. But there's no de novo docking implemented. Yep, Michal.
Okay. Any other questions?
Yep, we have Michal, I think.
Did you have a modification of bases, methylation or any modification parameterized?
No. Unfortunately, the biopolymers in MMB are all canonical amino acids or nucleic acids. There are no modifications currently supported. The thing is that it's not quite as simple to implement modifications or arbitrary ligands in internal coordinates because you have to set up this, this internal coordinate tree. And I'm planning to do that at some point, make some sort of interface that will let you specify that sort of thing. But at the moment, no, it's not implemented.
So at this moment, you can't really simulate ligands, small molecule ligands that are not yet parameterized in MD.
That's correct. I mean, unless your ligand happens to be like ATP or something, or an ion or water, or another protein.
Okay. Any other questions? So so how easy it is to actually take the the molecule, such as the ribosome, and parameterize it to describe it in terms of rigidifying some parts, making the other parts flexible. Because obviously you mentioned the time required to run the simulation itself. But how long does it take to write all the scripts? Because I'm familiar with various types of software that use restraints, and sometimes writing the script to run the simulation takes like 10 times longer than the simulation itself. Right.
I think that probably applies to MMB as well. So
how long did it take you to write scripts?
Let me tell you, it's a lot longer than, well, actually maybe it is 10 times the compute time. I don't know. I mean, if I had worked on it full time, maybe a month.
You presented adding the constraints as a positive thing, but did you encounter any examples where adding the constraints made the function you optimize harder to optimize because it got stuck in local extrema?
I have had constraints cause more problems than they solved. That what you're pointing out is actually that happens. But there's another thing that also happens, which I didn't fully appreciate when I started making some of these models, and that is that the way the constraints are applied in internal coordinates is very inefficient, and so I found that it was the number of the amount of compute time that you need to calculate the dynamics of your system rises in a very nonlinear way with the number of constraints. And recently I just started playing with that, and I can now actually have some molecules that are not even constrained, that just don't have any degrees of freedom even with respect to ground. And that saved scads of computer time. Like with a ribosome simulation, with a virus simulation that we were doing, it was like a factor of 8 faster, just because I got rid of these constraint equations for each protein in the capsid. In terms of constraints causing traps, yes, that's also an issue. A real molecule would have flexibility, which lets things maybe slide past each other easier than if everything's made out of rigid blocks. And that's an issue that's a real that's something we do have to confront when we're generating a model. I still think it's a lot faster to make these simplifications than to try to model these systems at all atoms resolution, or rather, not all atoms resolution, but with all atoms kinematics.
Okay, I have another question related to the previous one about restraints. So let's imagine that you, in many cases, in real biological experiments, you may obtain restraints for different states of the molecule at the same time, before you realize that the experiment actually looks at different states of the molecule. So for instance, one molecule exists in 2 different conformational states, and the experimental data, the restraints that you obtain, is actually a mixture of information about these 2 states. So how resistant is your method for restraints that are not fully compatible with each other? So let's say you have you try to run a simulation with 2 restraints that are to some extent mutually exclusive. Would it get stuck somewhere in between, or will the simulation lead for the system to jump back and forth between the two minima defined by different sets of constraints? So have you encountered anything like this?
So both of those things can happen, and so at the end of the day, garbage in, garbage out, right? If you don't actually know what your system is doing, and you have constraints that are in conflict, you won't ever be able to satisfy both. And you have to be, I think, a little bit smarter about what you do. I mean, I can't. There is no generic catch-all answer, but I do know that I've encountered this problem lots of times. For instance, let's say you're doing some maybe some cross-linking experiments, and you find that you have this monomer and it's tetramerized, but you don't know if it's tetramized in a symmetric way, or if there's symmetric dimer and then the dimer is dimerizing with the other dimer. Sort of prizing that information apart is hard. I'm afraid I don't have a generic answer, but I can tell you that forces are additive. So you can put forces that can't all be satisfied simultaneously, and they won't crash. I can't guarantee that you'll get the right answer though.